Hello everybody, so today I'm doing my Science Baccalaureate presentation on the potential and future of gene editing and gene therapy. Now this is a very large field with a lot going on in it right now, but I hope to give you guys just a good overview of what the field is all about, some of the stories that have arisen from the field in the last couple of years, and the potential as to where this type of technology can go. But before I get on with the rest of the presentation, let me just give you guys a little bit of a quick explanation as to what the difference is between gene editing and gene therapy. And they essentially are the same thing, but there is one minor difference. With gene editing, a mutated gene is either removed, replaced, or modified, where with gene therapy, a mutated gene is removed and replaced with a new and healthy version of that gene. Hello everybody, so for this slide, I'm going to have my face cam on, just to let the voice match the face, and I'm going to give you guys a little bit of an introduction as to who I am. So my name is David Campbell, I'm 17 years old and I go to the Anderson High School in Lerwick. And I don't know if you can tell by my accent, but I'm actually not originally from Scotland, but I originate from the deepest depths of the planet, known as Cape Town, South Africa. And you're probably wondering, David, what's the reason for you choosing gene editing as your topic? Well, 2 years ago, I was diagnosed with an eye condition known as keratoconus. And essentially what happens with keratoconus is that your cornea starts becoming more of a cone shape. So this is essentially leading to vision loss in my right eye, but it will only happen over a period of time. So it's a type of thing that's not going to happen right now. But I've luckily had treatment for my left eye, but I still need treatment for my right eye, which I can hopefully get soon. But this got me thinking, many mornings in the shower before school, online school, mad. and I'm worried that my future kids will end up picking up this disease and my grandkids. So this kind of led me down the rabbit hole of looking at gene editing as a way I could treat this disease. And this all kind of coincided with my baccalaureate teachers giving the baccalaureate students the task of choosing what topic to do our presentation on. So I decided to choose gene editing as my topic. Now, if you're interested whether I can treat keratoconus, my eye disease, with gene editing, I sadly can't. It's just bad luck, and most eye conditions are not genetically passed on. But there are a few exceptions to the rule, which I will get onto later. So let me get the final point up. I have terrible eyesight. So as I said earlier, it's a large field with a lot going on in it right now. And there are a lot of developments that are happening in this field lately. But one of the main developments that has come out of this field in the last couple of years is the development of the CRISPR gene editing technology. Now, the CRISPR technology is a new and unique way of editing the genome, and it's causing a lot of excitement in the scientific world. CRISPR actually stands for Clustered Regularly Interspaced Short Palindromic Repeats. So I've got it up on the screen there, all spelled out for you guys. So you can understand why we stick to the abbreviation CRISPR. It's a lot easier to remember. And there are actually different CRISPR systems that can be made. And one of the most well-known CRISPR systems that can be made is known as CRISPR-Cas9. And CRISPR-Cas9 is actually causing a lot of excitement in the scientific world because it's currently the most versatile and precise method of editing the genome. So the development of CRISPR-Cas9 was really pioneered by these two women known as Jennifer Doudna and Emmanuelle Charpentier, and they subsequently won a Nobel Prize for their work of pioneering the development of CRISPR-Cas9 with their research teams. I've actually got a bit of a funny story, but at the beginning of the project, when I was looking at people to contact and maybe carry out an interview with, I actually contacted Jennifer Doudna and Emmanuelle Charpentier. Now, Emmanuelle Charpentier, she left me on red. She aired me. She didn't get back to my emails. However, Jennifer Doudna, she didn't get back to me personally, but she passed me on to her assistant. And through her assistant, I managed to get in contact with Adam Wade Vallance, who's a PhD candidate at the University of California in San Francisco. And I actually managed to carry out an interview with Adam, which I'll get onto later. So I just thought that was a funny story. So in the next slide, I'll be showing you guys a quick video as to how the CRISPR technology works, just to give you guys a better overview as to what I've been speaking about.
This is DNA. It's the blueprint for making living things, from single-cell bacteria to plants to animals, including to humans. It influences almost every part of your physical being. The shape of your nose, the color of your eyes, sometimes your likelihood of getting certain diseases. But what if science could redraw your body's blueprint? What if it were possible to edit your DNA? Insert new sequences that change inherited traits, or remove genes that cause disease? Well, now it is more possible than ever with CRISPR. CRISPR is a combination of a scissor-like protein, such as one called Cas9, and a guide molecule. The molecule acts as a bloodhound, taking CRISPR on the hunt to specific sites within the genome. Once there, the protein cuts the cell's DNA on the spot. That completely disables the targeted gene. The cells then repair their DNA. They put in a new DNA sequence that the bloodhound also carried.
So I hope that video gives you guys a good general overview as to how the CRISPR technology works. But in this slide, I'm going to get more in depth and talk about how CRISPR-Cas9 works. So on the top right-hand corner of the screen, you can see the guide RNA. The guide RNA searches and binds to the target sequence. The Cas9 enzyme then binds to the guide RNA. So the Cas9 enzyme is in the middle of the screen there, and it's the big purple beam thing. It's the only way I can describe it, I'm sorry. Both strands of the DNA are cut by the Cas9 enzyme. Then, the cut is repaired by introducing a new sequence of donor DNA. So the donor DNA is on the left-hand side of the screen there. However, some scientists are still analyzing what happens if they cut a sequence of DNA and don't insert a new sequence. And this is just incredible to me, and this just shows why this technology has got so much potential and why it's causing so much excitement. However, there are risks of using CRISPR. Now, last year I carried out an interview with Adam Wade Valence, who's a PhD candidate at the University of California in San Francisco. And what Adam told me is the main risk of using CRISPR is that it won't cut the desired gene. So what does this mean? Well, it means that this may lead to some off-target effects. And some of these off-target effects may lead to some serious diseases, including cancer. So how do we solve this? Well, we need to regulate and test this type of technology to a point we know it is safe to use. However, although there are risks of using this type of technology, there are more benefits right now than risks. And one of the main benefits is that this type of technology is being used to make significant developments in terms of treatments for genetic diseases. Now, Editas Medicine is a discovery phase pharmaceutical company based in Cambridge, Massachusetts. And they are a company which I came across while doing research into this field, into gene editing. So I decided to give them a slide and give them their own little place in this presentation of mine. But they are currently working on multiple CRISPR treatments based on the CRISPR-Cas9 gene editing technology. And as you can see in the diagram there, they are well funded with $163 million worth of funding. And there's another company below them known as CRISPR Therapeutics, which has got $162 million worth of funding. And I'll get on to CRISPR Therapeutics a little bit later. But this just shows why this field is making so many developments. It's because these companies are so well funded. Now, Editas Medicine is using the CRISPR-Cas9 gene editing technology to develop a treatment for Leber congenital amaurosis, which is an early form of childhood blindness. You see, there are specific mutations that are at cause for Leber congenital amaurosis. So it's not that difficult for CRISPR-Cas9 to go and target this mutation and edit it. Also, your immune system has limited activity in the eye, thus the CRISPR technology won't bring any severe off-target effects. However, this is not the only genetic disease that Editas Medicine is looking at treating with CRISPR. Another one is muscular dystrophy, cancer, and even various blood disorders. Now, CRISPR Therapeutics, the company I mentioned earlier, is now working with another company known as Vertex Pharmaceuticals. And these two companies are working together to create treatments for beta thalassemia and sickle cell disease, which are types of blood disorders. They've actually successfully treated patients with beta thalassemia and have successfully treated one patient with sickle cell disease. You see, both these conditions affect the production of beta globin, which is a component of hemoglobin. Thus, disabled beta globin is produced. So you're probably wondering, how does CRISPR-Cas9 fix this? Well, CRISPR-Cas9 is used to edit a region of genes to disable the production of fetal hemoglobin. And the idea is that the fetal hemoglobin gene can be turned back on in the adult red blood cells. The fetal hemoglobin is meant to make up for the disabled beta globin. So it's meant to compensate for the disabled beta globin. And to me, this is incredible. The fact that this type of technology is even being used to consider to make treatments to certain diseases which seemed incurable 30 years ago is mind-blowing. But how far should we take this technology? And does it get to a point where we as the human race start to play God? And what happens if someone with a different agenda decides to get their hands on this technology? Well, an example of this happened with He Jiankui. In 2018, He Jiankui announced that him and his colleagues were able to genetically modify the genes of 2 babies to make them resistant against HIV. Whether his claims are true or not that these babies are resistant against HIV, we don't know. But what we do know is that he did actually genetically modify the genes of these 2 babies. Now, He Jiankui claims to have disabled the CCR5 genes of these 2 babies to make them resistant against HIV. Now, the CCR5 gene is a gene that encodes for a protein that allows HIV to enter immune cells. However, disabling the CCR5 gene does more harm than good, as this gene can help you fight off the effects of some serious diseases and viruses, such as the West Nile disease and even the common flu. Luckily, He Jiankui and his colleagues were fined quite severely and sentenced to time in prison. Now, nothing will ever take away the negative stigma He Jiankui and his colleagues have given these babies, a stigma that they are not natural and they are artificial. And to me, this could become a huge issue, especially if gene editing becomes a common medical practice in the future. Luckily, there is a solution to this, and it's a natural one. The stigma will lessen as the public does become used to this type of technology. However, this isn't the only ethical concern gene editing may pose. Another one is that this type of technology may lead to greater social inequality, as only the rich will be able to afford it and the poor won't be able to. I mean, some of this technology will cost up to $1 million, which only the rich will be able to afford, as I said, and it will give them an even greater head start in life. So how do we combat this greater social inequality that may be caused? Well, maybe not allowing the physical features to be edited and only making the babies be allowed to be edited to make them resistant against certain diseases and genetic diseases. However, this will still lead to greater social inequality, but probably not as much if the physical features were to be edited, because that would lead to new forms of racism. And in 2021, we don't need that. We're already dealing with what we have now, so we don't need new forms of racism. And to be honest, I have many questions about the risks gene editing may pose, especially if it becomes common medical practice in the future. I mean, is it really worth editing the genes of healthy people to avoid unhealthy people being conceived? Well, last year I sent a couple of questions to David Resnick about this, who is an American bioethicist at the National Institute of Environmental Health Sciences. And what David told me is, well, he gave me a bit of a generic answer. But what David told me is that we as the human race should proceed with caution to minimize harm. And who knows what the effects may be if we go full throttle ahead with this type of technology without looking at the risks and side effects. And to be honest, I completely agree with him. You know, it can get to a point where we as humans start to play God. I mean, some of the worst case scenarios gene editing may pose may lead to greater social inequality and a negative stigma being attached to people because they're genetically modified. I mean, this just shows how out of hand this technology may get. However, it may get to a point where we as humans can possibly give ourselves wings, which I think is actually kind of cool. But seriously, we do need to be careful because we can start to move out the realm of being human and become something else. So I want you guys to think about something as I end this presentation. What does it mean to you to be human? Now, thank you guys. That's my whole presentation on the potential and future of gene editing. But on my last slide, I would just like to thank people that have helped me throughout this presentation. So I'd like to thank Adam Wade Valence, the PhD candidate at the University of California, who I carried out my first interview with. I would like to thank David Resnick, the bioethicist at the National Institute of Environmental Health Sciences, that I sent a couple of questions to, and he actually got back to me. So I'm grateful about that. I'd like to thank Mrs. Reed and Mr. McGinley, my science baccalaureate teachers, for helping me with my feedback, giving me feedback on this presentation, and just helping guide me through this whole presentation and this whole process of getting this thing put together during this really weird and difficult time that we are in right now. And I'd also like to thank my mom, my dad, and my brother for just giving me feedback on my presentation and telling me things that I could do with it and how I could improve it. So thank you, everyone.